Friends, today we are going to see the processing of rRNA, and in our previous video we have already discussed about the overall view of the RNA processing. So the newly born RNA primary transcript are processed both in eukaryotes and the prokaryotes to generate the mature and active rRNA. The mammalian transcript is a 45S RNA molecule which has on one copy each of the 28S, 18S and 5.8S. The rRNA transcript undergoes the following two changes before its processing. It is completed into following two steps. That is, first step is that to each transcript, 110 methyl groups are added to the ribose molecule, and these methyl groups are retained in the mature molecules. Thus, the ribose methylation seems to act as a marker for the cleavage sites. So, the first processing reaction seems to be a cleavage of the superprocess extra leader sequence of the 5' end, and the next cleavage occurs within the spatial sequence located between the 18S and 5.8S sequence molecule. So it can be described by the figure, so we can better understand through the figure. So processing and modification of the rRNA, it requires a class of small RNA, that is snow RNA, that is small nuclear RNA, which is constituted of the 60 to 300 nucleotide sequence which are associated with the protein fibrolarin, an abundant component of the nucleolus. So snow RNA has two subtypes, that is C by D box and H by ACase box, that also known as the pseudo-uridylation. So, in the case of the vertebrate, the rRNA contains specified methyl groups, and in yeast rRNA, there are 43 pseudouridine residues which is produced in different steps. So, RNase 3 is responsible for the cleavage of the 16S and the 22S RNA. So, trimming reaction appears to be necessary for generating the authentic 5' and the 3' end. Finally, the rRNA sequences are preceded by the RNAP, also known as RNase P or RNase 3, and other processing enzymes. So in this figure, there is a sequence pre rRNA, and 18 as 5.8 as and 28 as subunits are there, and with the help of the chemical modification and the cleavage and trimming, that is methylation and the isomerization, it separates, that is 18 as 5.8 as and 28 as cleaved separately. So in the case of the E. coli, that is prokaryote, there is a 16 as in behalf of the 18 as and 23 as and the 5 as, and there are two steps, that is primary processing and the secondary processing. RNase P and RNase E by F cleave the three subunits, and the trimming is done by the RNase M. So it separates like this: 16S RNA, tRNA, 23S RNA and a 5S rRNA. So overall view of the processing of the rRNA. Thank you for watching.